Hello everyone. You have been listening to 3 weeks of lectures on computer-aided drug design. I hope you are enjoying the course. I thought in the next 1 hour of live session, I'll just give a rundown of what you must have covered in the past 15 lectures. And there are 1 or 2 important questions which some of you have raised. I'll talk about that as well. Okay. So as I mentioned in my course that drug discovery is a long process and it's an expensive process as well. Okay, so there are a lot of issues that needs to be looked at related to the drug likeness property and related to the animal studies and so on. So traditionally molecules were designed in the lab and tested in biological assays. When they were found good, they went into animal studies and then later on human volunteer studies. And many of the compounds failed because they did not satisfy some condition or the other. Maybe it was not good aqueous soluble, or maybe it was not stable at the pH. So maybe it was not passing through the lipid membrane going into the bloodstream and so on actually. So many compounds failed during these situations, okay, and hence what happened is a lot of money was lost and time was also lost. So it's very, very important of late that as you look at new molecules which shows activity, that is lead molecules, you simultaneously also look at those properties, okay, the drug likeness property, the absorption, distribution, metabolism, excretion property, toxicity properties, side effect properties, and so on actually. So simultaneously not only leads were discovered, the leads were also optimized. That means they found a very good active molecule, but maybe the solubility was very poor. So you need to optimize, improve the solubility. Or maybe it was not getting absorbed through the GI. So you need to improve the lipophilicity and so on. So lead optimization was going on simultaneously. Okay. So that when you go into preclinical and clinical trials, you don't get surprises because where there is a lot of expensive trials where clinical trials are, and the drug company loses a lot of money as well as time, especially if a molecule fails because of some other reason. Okay, so now a lot of thought is being given in the area of in silico drug discovery, okay, where you try to identify the target for a particular disease. For example, if I'm interested in inflammation, should I go look into the leukotriene pathway or the prostaglandin pathway? Do I look at COX-2 enzyme, or do I look at prostaglandin E synthase enzyme? Like that. So the target you decide. And then once you decide on the target, you look into the lead. This is my course is generally covering this portion: lead identification, lead optimization, drug likeness property. So we use, that means when we talk about drug, generally small molecules, which goes and binds to the target. So the focus of this course is generally here. Can we use computational tools to sort of predict some of these properties? Of course, ultimately you need to do a wet lab validation, but can we use some computational tools? There are nowadays a lot of computational tools available to calculate many properties. Okay, so can we use them simultaneously? So drug likeness, toxicities, side effects, absorption, distribution, metabolism, excretion, and so on. So we can screen large number of molecules, and if you are not happy with some of them on the computer screen, you can eliminate them. You don't go into a wet lab at all. Okay, so computers can be used for target identification. Okay, we can identify the targets using proteomics, bioinformatics tool. Then structure prediction, 3D structure Then we can predict the active site of the target. Then we, of course, go into the lead identification, lead optimization, using a large number of tools. And then we can predict properties, toxicities using quantitative structure activity relation, quantitative structure property relation, quantitative structure toxicity relation. I think you will be coming across all these in the next set of lectures. We can use pharmacophore based modeling, try to identify what are the common features between certain leads. We can even predict degradation product. Okay, so many things can be done using computational tools. So if some of the properties are not good, we don't proceed with those molecules. We pick up some other molecule, or we try to modify the structure and see whether the property improves, and whether the activity improves based on structure activity relationship. Then we pick up and go into the wet lab validation. Okay, so a lot of applications where computers can help in the drug discovery process. Okay, so that you don't come across a lot of surprises as you go down in preclinical and clinical trials. Okay, so a drug is not only activity. That means activity alone is not enough, but so many other things have to happen, right? Because it has to go to a single target. We don't want it to bind to many targets and cause side effect. For example, if you look at some of the NSAIDs, non-steroidal anti-inflammatory drugs, they will go and bind to many enzymes in the prostaglandin and leukotriene pathways. So there could be side effects, oral absorption, because most of the drugs we take orally. We would all like to take drugs orally, not through intravenous or intraperitoneal or intramuscular. So how good is the absorption? That means it should have good solubility in your stomach, and it should cross the lipid barrier and reach the bloodstream. Distribution inside. Once it goes inside, it gets totally distributed into the bloodstream. So it is like putting a spoonful of salt in a bucket of water. The salt gets distributed, concentration goes down. If the volume of distribution is low, the concentration will be high. If the volume of distribution is large, concentration will be less. So for these depending upon the molecule, the volume of distribution can change. For example, if the drug is very polar, it will remain in the bloodstream. If it is lipophilic, it will not only be present in the bloodstream, it may get distributed in the lipid fat. So the volume becomes large. If the molecule is very, very small, like ethanol, it can go and distribute in interspecies. So the volume of distribution could be very large. Metabolism. Liver is a very fantastic gatekeeper. So it degrades through phase 1 and phase 2 biotransformation. In the phase 1 biotransformation, hydroxylation takes place, or oxidation takes place, or reduction takes place. So your drug or xenobiotic or any molecule gets either oxidized or reduced or hydroxylated or deaminated. And then in the phase 2, it puts in sulfates or glucuronide. So the compound becomes hydrophilic or polar. So the drug may get, the compound may get excreted either through urine if it is very hydrophilic, or through the biles and excreted if it's a bit hydrophobic. So drugs can get biotransformed. So you don't want that sort of metabolism taking place, because then drug will not be reaching its target. Then we have the excretion. Excretion is we don't want the compound to remain forever in the body. It has to come out. That means it should have certain half-life. Once it has done its job inside, it should come out. Otherwise, you may have a slow buildup of the drug, and over a period of time, we don't know what will happen. The long-term toxicity issues. Okay. Then, in addition, we have other solubility at the pH 2 and 7, because if you look at stomach, it's extremely acidic. It's got a pH of 2, and as you go down into ileum and then jejunum, and then you go to your colon, that's the large intestine, pH becomes basic. If it goes to the bloodstream, again pH is 7.4. So your drug is to be soluble in all these pH conditions. Otherwise, it can precipitate out. We don't want that. Then something called bioavailability. That is, bioavailability is how much the drug you take orally and how much it reaches the target site. That ratio multiplied by 100 is called bioavailability. So because of so many effects that the drug encounters on the way, the concentration of the drug in the target site may be so little when compared to how much drug person is taking, so the bioavailability could be very low, and so the patient has to take a higher dosage of drug, which could lead to local toxicity. So if the bioavailability is large, then you take less amount of drug, so local toxicity is less. So some drugs, for example, metformin has 74% bioavailability, whereas drugs like Taxol, which is an anti-cancer drug, may have 25–30% bioavailability. Okay, so bioavailability is a very important parameter, and then no side effects. We don't want the short-term side effects. We don't want the long-term side effects, and so on. Actually, okay. And then, of course, no toxicity, because the drug should not have any toxicity, whether it's genotoxic or nephrotoxic, and so on. Okay. There is a question which says, what is affinity? Affinity is if it is going and binding to a particular target, that tells you affinity. Okay. Efficacy is how effective is that drug based on what is the concentration you take. Potency also is somewhat related to efficacy only, but potency is what is the later effect that is found after the drug has been taken. It is called the potency, actually. Okay. So in addition, the metabolites also should not be toxic, because we are talking about a drug which gets metabolized in the liver through phase 1 and phase 2 biotransformation. So there could be a lot of side products which are formed. That also should not be toxic. Then we have the stability, pH 2, because we have drug present in the stomach at pH 2, drug present at various pH condition, as well as inside the plasma where the pH is 7.4. So it has to be stable at all these conditions. Then there is a barrier that's called blood-brain barrier. It separates the brain from rest of the body. It allows through active transporters only the nutrient like glucose. It allows oxygen inside, CO2 outside. It prevents transmitting of metabolites. It prevents transmitting of viruses, bacteria, xenobiotics that are present in the body. So it maintains the brain completely free of whatever other chemicals, compounds, neurotransmitters, viruses that are present in the rest of the blood region, body region. So you don't want your drug to cross this blood-brain barrier, okay? Because, for example, I am taking a drug for a pain, inflammation, and we don't want that drug like ibuprofen going into the brain. We don't know what will happen to the brain region because of the movement of this ibuprofen into that. Then there are a lot of P-glycoprotein efflux transporters, okay, P-glycoprotein, which will sort of efflux out your drug. These PGP efflux pumps are meant to throw your xenobiotics out, but you don't want your drug also to be caught by these PGP efflux transporters. And if it starts throwing them out, you are going to have lower concentration of the drug available. So its availability goes down quite a lot, right? So we don't want our drug to get thrown out by these PGP efflux transporters. So we need to design the drug so that it does not become a PGB efflux substrate. Okay, these efflux proteins have an active site where if the drug gets caught inside the active site, they can get thrown out. Okay, so then of course shelf life is also very important, because drugs have to be stored, and by the time it goes to the market, goes to the patient, it may take a few months. And then which form it has to be given, whether it's going to be oral, intravenous, intraperitoneal, intramuscular, nasal, anal, vaginal, dermal. So if it is in a liquid form, you may have to add other solubilizing agent, and then if it is in a solid form, it has to have some stabilizers, it has to have some binders. For example, when you take a tablet, it has a binding agent, it has got a coloring agent, it has got some stabilizing agent, whereas in the liquid form, there will be some surfactant which will keep the drug in either emulsified form or totally soluble form, and so on actually. So this is also very important. Normally, when drugs getting approved either through the FDA, that is Food and Drug Administration of US, or Drug Control Authority in India, the company is also mentioned in which form they are planning to give the drug, and they have to get approval. So for example, if they get approval for oral, that doesn't mean they can sell drugs for IV or IP. They have to again get approval for IV form or IP form, because the other constituents that are added are very different. If it is a solid drug, or is it IV or an IP. Okay, then mechanism of action. Nowadays, there is a lot of interest that you need to tell which proteins it goes and binds to. That's very important. We can, in olden days when drug discovery was performed, they just looked at whether the drug reduced fever or the drug reduced inflammation. But now we need to tell it is reducing inflammation, but at the same time it is binding to these enzymes present in the prostaglandin pathway. So FDA expects you to tell the mechanism of action. And of course, cost and ease of manufacturing, those factors come in, because these are engineering related issues. Okay, so a lot of things, as you can see, we need to have. So many nice looking compounds which are very active in your lab do not work well as a drug when it goes to clinical trials because of these problems. That is why nowadays there is very importance given to the drug likeness property or lead optimization. So as a chemist, a medicinal chemist or a synthetic organic chemist, he or she has control on this. They can make new molecules with more hydrogen bond donors like OH group, NH group, and more acceptors. They can change log P. So a chemist can do all this. But by doing this, or ester bonds, they can put amide bonds, they can make it more crystalline in nature, they can make it into salt form, chlorides, or potassium salt, and so on. These in turn will affect these properties. Okay? These in turn will affect all these properties, but it is not that it will affect one-to-one. That means when I change, add a OH group, it is not that it will change only one parameter. So they will also have contact with each other. First, melee. Oh. Next slide. So it is not that there is a one-to-one relationship between structural feature and its behavior. Okay, so for example, if I add a OH group, it may change the water solubility because it's become more hydrophilic, but it may change the permeability through the GA also, because it's become more hydrophilic. So one change here in the structural feature can have 2 or 3 changes. That is why the whole drug discovery is extremely challenging. Okay, so for example, if I put a ketonic group is nothing but C double bond O. Okay, so I am making it a little bit hydrophilic. So by doing that, maybe I am improving the water solubility. Whereas by doing that, I may be affecting its other properties like absorption. Okay. When I put an ester bond, I am making it more water soluble because it's becoming hydrophilic. But when I put the ester bonds, metabolism can happen because there are enzymes like esterases, lipases present in our blood and liver, which may degrade this bond. Okay. By making it crystalline, I may be reducing the water solubility, but I may be improving the stability of the molecule. Okay, by making it amorphous, I may be improving the water solubility, but it may be getting more degraded. Okay, so many things can happen. Okay, but as a chemist, if a chemist is interested to be in the drug discovery, he or she should know how these features connect with these properties and how they can play around in order to modify certain properties here. Okay, that's what is called lead optimization. Okay, so we can optimize the solubility, we can optimize the stability, we can optimize the GA absorption, we can optimize the bioavailability by doing all these things which a chemist knows better. Okay, many drugs fail because of this. Okay, as you can see, poor ADME properties: absorption, distribution, metabolism, excretion. Lack of efficacy. Okay, lack of efficacy means it's not acting. Like somebody just now asked, what is this? It is not acting as it is supposed to. Okay, because maybe poor bioavailability. It should, an antibacterial drug is not acting. It's because not that the drug is not good, but maybe the concentration available at the target site may be less. Okay, so it is not as efficient. Toxicity. Maybe the drug is toxic. Short-term toxicity, long-term toxicity, nephrotoxicity, genotoxicity. So many types of toxicities may be happening. So you see, about 90% of the drug when it goes to the clinical trials fail because of all these problems. Okay, 90% or even much higher. That means pharma companies lose a lot of money. Nowadays, FDA expects many things from the pharma companies to produce. So 95%, 90% of the drugs which enter the preclinical, that is animal studies followed by clinical, phase 1, phase 2, phase 3, lose because of these problems. So if computational techniques can help in predicting a priori so that we don't consider all the molecules which are very active in the lab, but we take activity as one part and the other drug likeness properties on the other part and sort of balance. So the best compound may not have the best drug likeness property, whereas the second best compound may have. So this is what it is. You have poor ligand, very good ligand. But then properties are so bad, so it doesn't go as a drug. Okay, you may find a nice compound in your lab. Okay, we will call it a lead molecule. We may call it a hit molecule, but then it's got completely poor properties. Okay, so we have a good drug. Okay, the properties are good. Good ligand, good properties, so it can be a good drug. Little bit poor properties, poor activity, but then still the properties are good. Even this can be a drug. Okay, so ideally you may like to be here, but you will never achieve this. You may achieve like this. That means good activity and reasonably good property, or even slightly lower activity, but again good properties. Okay, and that is what you are trying to do in lead optimization and so on. Okay, so this is also an important slide. You must have seen this. So how the drug keeps traveling down, down, down. When we take an oral drug, solubilization, GI penetration, otherwise it gets excreted, goes to the blood. Then this is called first pass metabolism. Okay, and then goes. Sometimes drugs get deposited in the tissues, so some concentration is lost. Okay, some concentration is lost, and then so when it gets metabolized, it goes into kidney, urine, lungs, exhale, and so on. Then some of it goes, and then some of it may go to other sites, unwanted side effects, and then only some of it goes to the desired biological activity. So what we take orally vis-a-vis what is really reaching, okay, that is the action of the drug. And we may think I'm taking so much, it's not really effective because of all these reasons. Okay. Another important point we need to consider is the changes in the pH, as well as the okay, there is a question, drug repurposing. For example, imagine there is a drug. A fantastic example is aspirin. Aspirin originally was started for fever. Then what happened was aspirin is now being used for cardiovascular. If somebody has a cardiovascular problem, for blood thinning, they are saying aspirin which can be used by people who have cardiovascular issues. They are asked to take aspirin once a week or once a month. In fact, in US, it's being suggested that everybody above the age of 40 or 45 are asked to take aspirin once a month. So that is repositioning. And now there are some studies where aspirin is being even looked at as a which can work towards certain types of cancer. Okay. Similarly, if you look at metformin, it's an anti-diabetic, but there are some studies which show that it can have some anti-cancer properties. So there is now a lot of interest in looking at drugs which are already approved for certain diseases. Can it be working on some other diseases? Okay, that is what is called repurposing or repositioning. Other one is targeted drug delivery. For example, imagine I have a drug. It's got a very bad lipid permeability. So can I encapsulate it using some material, some polymer, which will allow the drug to cross the GI, and then it goes into the bloodstream and goes directly at a certain target. Suppose I am targeting a tumor. So can it go directly to the tumor and then release the drug? So that way my bioavailability of the drug will be very high, because a normal drug, like I said, it goes to the bloodstream, it gets absorbed in the tissues, it gets degraded in the liver because of phase 1 and phase 2 biotransformation. So if a drug can be targeted directly to go and go to a set of cancer cells, and then it releases the drug, then you get very large quantity of the drug directly targeted to the site of interest, like a tumor. So, for example, it has been found that some tumor cells have certain proteins on the surface. So, can I have a targeted drug delivery which can identify those surface proteins, so they will directly go and attach to them and release my drug. There are some targeted drug delivery systems which can be based on thermal. For example, it's been found that certain tumor may have slightly higher temperature than the rest of the body. So when the drug encapsulated reaches those tumor sites, because of a slight increase in temperature, the drug delivery system is able to release the drug at those sites. In fact, there are some studies which is being done on using some magnetic nanoparticles. So I have the drug encapsulated in magnetic particles. So using a magnet, external magnet, whether those encapsulated drug can be driven to certain locations and then release the drug. That is more in the theoretical region. So targeted drug delivery is designing a system, encapsulation system, which will overcome some of the barriers and reach your location like a tumor or an inflammatory site, and releases the drug so that the bioavailability of the drug is very high. Okay, let's continue on what we're talking about. So if you look at your GI, especially the stomach, the small intestine, and the large intestine, or the up to the colon, the pH is very, very acidic in the stomach too, whereas it becomes basic as you go down right up to your colon. And also the surface area also the pH difference is extremely acidic in the stomach, and it becomes basic as you go down. So a drug which has slightly acidic or slightly basic, that is pKa in the acidic region, will get generally absorbed in the stomach, whereas if you have more basic drugs, it can get absorbed right down. And that is one point. Another point is the surface area of your small intestine is much higher than the surface area of your stomach. So absorption, mostly it is a passive absorption. So passive absorption is much higher in your small intestine when compared to large intestine. Okay, so this. So you need to understand the surface areas, the pH, so the absorption of drug in different locations. Now the transit time is also very different. As you can see, the transit time in the stomach, that means the stomach emptying approximately happens in about 3.5 hours, whereas if it go down in a colon, it's 1 to 3 days. Another interesting thing that happens is the pH changes dramatically if it is an empty stomach or an empty jejunum or a colon as against fed state. That is why sometimes doctors tell you take the drug before food, take the drug after food, or take it after 2 hours, because the pH are changing so much between the empty state vis-a-vis the fed state. Some of the absorption patterns can change very dramatically. We need to understand these as well, because after all the drug absorption is a function of the pH condition, the surface area, as well as the transit time. Okay, the next point which we need to look at in addition to this is the oral bioavailability. This is the most important. For example, metformin, like I said, has very high oral bioavailability, so we take less of it. Whereas Taxol, which is an anti-cancer drug, which has very low oral bioavailability, that means you need to take more of that drug to get the same effect at the target site. So it could have local toxicity. So how do you calculate oral bioavailability? The dose we take, and then what is available at the target. Okay, that's generally oral bioavailability. Sometimes what they do is when they do it on rats, they give the drug orally. Okay, and then they also give it in intravenous, and then look at the ratios, because intravenous will have very high concentration, whereas if it is on It will be very low concentration if it is given as a oral, and then from that you can tell what is the oral bioavailability. So generally it is felt that about 35% oral bioavailability is good enough. I mean, it's good. Ideally, if it is higher, is good, but at least it should be 35%. That otherwise, you need to take larger concentration of the oral bioavailability. Okay, so lipid solubility, like I mentioned, weak acids, weak bases get solubilized in the stomach because your stomach pH is 1.4 to 2.1. So aspirin. So when you take aspirin, immediately you get effect, right? Because it's acetyl salicylic acid. So it's got a pKa or low pKa. So it's getting absorbed in the stomach region, and quickly you get a relief. Whereas if it pKb, then you are not going to get absorbed in the stomach. The absorption may take down the line. So salicylic acid, aspirin, thio, pentanol, all these type of antipyrine, they're undissociated, so they get absorbed in the stomach. Whereas if you look at basic anti-malarial drugs, they are going to get absorbed not in the stomach, but it will happen much lower in the small intestine. Okay, so if you want to design drugs for quick action, make mildly acidic or maybe a little bit basic, so that it doesn't get dissociated in the stomach. So if it is not getting dissociated in the stomach, then the drug will be in the neutral form. If it is dissociated, you are going to have the anionic and a cationic form. So then it's not going to get absorbed because they are polar. And as you know, your stomach lining is highly lipid. So the drug should not be in the dissociated form if you want the drug to get absorbed in your stomach. Okay, if it is in dissociated form, then it will not get absorbed in the stomach, but it will get absorbed down. Okay, so that's another important point we need to consider when we talk about the quick acting drug and slow acting drug, as well as the pH conditions between the stomach, ileum, jejunum, and the colon. Okay, the next important point we need to consider is okay, so as you can see, the drug discovery activity is one aspect. So the activity is one, but the issues of the physicochemical properties of the molecule and what the human body does to the drug, that is the pharmacokinetic properties, are also very, very important for us to understand. Okay, so in addition to then once it gets absorbed, there is something called distribution. It is like putting a spoon of salt into a big bucket of water, so it gets distributed and concentration of the salt will be certain number. Okay, so that's the distribution. Distribution is important because it tells you what is the maximum concentration of the drug that it reaches in your bloodstream. Okay, whether that is effective concentration or whether it is a toxic concentration. This volume of distribution varies depending upon the type of drug, because if it's very hydrophilic, then it will remain in the blood plasma. If it's a hydrophobic, it can go into the lipid, so the volume of distribution increases. Okay, whereas if it is very small molecule like ethanol, it can have very large volume of distribution because it can go in any place it wants. Okay, so volume of distribution is important because it tells you how much concentration of the drug available at the target site. Okay, so this volume changes depending upon the race. For example, anti-malarial drugs or even anti-tubercular drugs are not toxic to Asian population, whereas they could be toxic to the Caucasian type of population, and so on actually. Okay, so volume of distribution we need to understand. Then in the there is a lot of biotransformation taking place in the liver and in other parts of the okay, plasma and so on. So like I mentioned, there is a phase 1 transformation, there is a phase 2 transformation. So simple hydroxyl groups are put, or oxidation takes place, or reduction takes place in phase 1. And then in the phase 2, you are adding sulfates or glucuronides and so on. So you make your drug very, very hydrophilic, and hence it may get excreted from the plasma through the urine. Okay, so if somebody takes a drug through a oral route, generally you have a graph, concentration of the drug going up, okay, and then it goes down because of elimination. Okay, so if I am giving an IV type of drug, of course concentration will be very high and goes down, whereas if it is an oral, there is absorption and things like that, so the concentration may be much lower. So this ratio we will call it as bioavailability. So what they do is, if they are going to test on animals, they may give the drug orally and then monitor the concentration as a function of time. The x-axis is time, and then they will give the drug intravenously and monitor both as a function of time, and then area under the curve oral versus area under the curve intravenous into 100, that gives you the percentage bioavailability. Okay, so that is how it is done. Okay, we can model these, mathematically model. When you go down to class 38, 39 lectures, you will come across these. Okay, okay, then we can calculate many pharmacokinetic parameters: volume of distribution, plasma clearance, plasma half-life, bioavailability, using these graphs. Using these graphs, we can calculate all these. I think you must have come across. I've talked about it over the period of time. There are many rules for bioavailability. I think I talked about Lipinski's rule of 5. This is a very traditional rule which says molecular weight less than 500, log P less than 5, number of hydrogen bond acceptors less than 5, okay, less than 10, and then donors less than 5, and so on. Of course, as I said, rules doesn't mean it has to be strictly followed. There are many drugs which don't follow the rules, and there are many molecules which follow the rules but still can have a poor bioavailability. And then there are rules like Weber's rule, Gose et al. Rule, Newgay rule. So many rules are there. So we can have softwares. Many softwares can give if you give a structure, like I showed you Swiss ADME, and then many softwares will tell you whether these rules are followed or whether these rules are not followed. So we can eliminate molecules which you think do not follow those rules, and take up those molecules which follow those rules, and then we can spend more time in the wet lab with those molecules. Okay, so these rules are very useful for that. And as you go along, you must have come across blood-brain barrier rules, and then later on we will look at PGB, Flexor rules, then GA permeability rules as against the polar surface area. Then there are Opera rule of 3, and then there are rules for certain cardiovascular protein binding. Like that, there are many rules we will come across. So if you design new molecules on your screen, we can check it out whether the molecules satisfy all these rules, and select only those which seem to satisfy all the rules, and then we can take them down into our wet lab. Either as a synthetic chemist, we can synthesize those molecules, and then as a biological chemist, as a biological scientist, you can perform the biological activity studies. Okay, so the rules are extremely useful in such a situation. Okay, so many rules are there, and as I said, when there is a rule, there is always exception. So there are small variations between the rules. For example, one rule says molecular weight should be less than 500. Another rule may say molecular weight should be between 120 and 450. So there is nothing like one single rule which is hard and fast. So you may get some variations between each one of them. You may get different variations between each one of them. Okay, so you may get different, but then we can use these rules to see which ones are most probable candidates which have good oral bioavailability and oral good ADME properties, and which ones might not have good properties. So we can bet on those so that we can select those which plays a good we can only select those actually. Okay, so many rules are there. And then of course toxicity, we should know the toxicity limits and how we play. So for example, activity or efficacy as increased concentration response. If you are looking at how the tumor size changes as I increase concentration. So this is called efficacy. Maybe at this concentration it is toxic. So we work here. But this is a very nice picture to have. But in many cases it may be very close. So little increase in concentration, activity increases. Little more activity is also increasing, but toxicity is also increasing. Okay? So many anti-cancer drugs have this problem. So they may have good efficacy, but they are also having toxicity. So we need to play between the ratio of toxicity versus efficacy. Okay, but ideally, if you get this type of situation, you are far away. Your toxicity limits are so far away from the activity graph. But as I said, this is a very theoretical, nice situation to have. But most of the time, their toxicity is also going to come closer, like anti-cancer drugs. So little more increase in concentration, activity also improves, but toxicity also improves actually. Okay, so we are going to talk about in the next the PGB efflux. That means whether your drug is going to have effluxed out through PGB. What are the various metabolisms? Plasma stability, because plasma contains a lot of hydrolytic enzymes. So your drug may have problems of hydrolytic enzymes. And plasma stability, that is whether the drug will have hydrolytic enzyme degradation. Plasma binding means your drug can get bound to human serum albumin. If you have organic acids like carboxylic acids or amines, they can bind. And then this is some drugs can affect the electrical activity of the heart. Okay, so we want to find out whether it will have any effect on the electrical activity of the heart. So many rules are there. Like I said, as I go along in the next few more lectures, you will come across all these rules actually. We will not, as I said, use many of these rules. Try to look at molecules which may be fully satisfying the rules. There may be molecules which may be totally not satisfying, but there could be molecules which may be satisfying part of the rules or near borderline. So in such situations, you may still consider them, and we may do some optimization to improve some properties. Maybe if it has got too many hydrogen bond acceptors, so one rule is not satisfied, but activity looks good, you may take that structure and remove the hydrogen bond acceptors. Like you have OH means we try to make it OCH3, or we try to block Like that we can still do based on your medicinal chemistry expertise. Okay, there are 2 or 3 things which I would like to talk about, which were raised in some questions past a few lectures. It is not that there is a very clear rule, especially with respect to rotatable bonds and okay. Rotatable bonds, there is no single rule at all. As you can see, I've taken from this reference, it says each set of definitions has its loopholes and problems. Okay, so the rotatable bond definition is any single bond not in a ring, not bound to a non-terminal heavy atom, that is non-hydrogen atom. Okay, you exclude CN bonds because they have high rotational energy. Okay, so in this particular case, we can call these rotatable bond. You can call these rotatable bonds. Here, this is a rotatable bond. 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 This one does not have a rotatable bond. Look at this. There are many exceptions. Each set of definition has loopholes and problems. Bonds that are not rotatable are non-single bonds. That means if you have double bond, triple bond. Bonds to hydrogen and other monovalent atoms like halogen, bromine, chlorine, iodine, that's not. Ring bonds, they are not rotatable. Bonds to terminal atoms including CH3, NH2, OH, they are not rotatable, sorry. Okay, so you cannot take this as a rotatable bond. Okay, the CN bond, carbonyl and amide bond, bonds connecting 2 aromatic rings with collectively 3 or more ortho substituents. So even though you may take this as rotatable, but if there are going to be some ortho substituted, then that is not going to be rotatable. Bonds connected to terminal triple bonds, including bonds to cyano groups. Okay, so if you have C triple bond N and then you have a single one bond, then that is not connected as a rotatable bond. So a lot of exceptions are there. Lot of exceptions are there in this rotatable bond. So there is nothing like a single rule, because when you say a rule, there is exception. When you say a law, then there is no exception. Like Newton's first law, there is no exception. But when you say a rule, there is always, like I said, Lipinski's rules, bioavailability. There are a lot of exceptions where there are drugs in the market which do not follow Lipinski's, but still are drugs. So in the case of rotatable bonds, this problem is there. Okay, so you need to understand a little bit of the chemistry like to do it. This example is taken from this particular reference. So this is a rotatable bond, and this and these are rotatable bonds. In this particular case, these are rotatable bonds. So rotatable bonds have it's not very straightforward. Another interesting thing is related to hydrogen bonds. Okay, again, hydrogen bonds are also not very straightforward. It is predicted based on criteria of directionality, because hydrogen bond formation is a directional, unlike electrostatic forces. It is not directional, whereas hydrogen bond is very directional. Okay. So generally, COO ester greater when compared to acid esters. Okay, then when compared to amide ketones, when compared to acids, C double bond S, and so on actually. And you can see that water at say at 0 degrees centigrade can even form 3.7 hydrogen bonds around each water molecule. But when it is at 100 degrees, it can go to 3.2. This is taken from this reference. So the hydrogen bonds that water can form can keep varying depending upon the temperature. Okay, so 3.2, it never forms 4 generally, then 3.7, then it comes to 3.2, and if it becomes water vapor, it keeps coming down. Okay, let's not bother about it, but it, at boiling point condition, it forms here. Okay, then you can see the group probability. So the probability of hydrogen bond formation can vary between various groups. As you can see, this probability can vary. High probability of 0.9 going down to very low probability also. So water hydrogen bond forming ability for various functional groups, hydrogen bond forming ability of various functional groups can change. As you can see, the probability can change from very high number 0.10, that means most probable, to even very, very low. Okay, so it's not very, again, hydrogen bond formation is not very straightforward because it is very directional. That means how the orientation of various functional groups in one compound, another compound is found. So that problem is also not very straightforward. So two things, one related to rotatable bonds. There is no single rule which tells if you do this, you can predict the rotatable bond, because there are so many exceptions which are found in rotatable bonds. And also hydrogen bond, because being it's directional in nature, how the bonds are, how the functional groups are present, or how the conformation is present of functional groups which can form bond, hydrogen bonds are present, that determines whether it forms a hydrogen bond or not. And as I showed you for water from this reference, it can change quite a lot depending upon temperature also. Okay. Then there was one more doubt which was asked in many of related to statistics and probability. This is based on a very simple your knowledge of statistics. So if you think we have three phase 1 clinical trials, phase 2 clinical trials, phase 3 clinical trials. So imagine 100 molecules are entering phase 1 clinical trial, and if there is a 60% failure, that means 60 of the molecules will not pass phase 1, only 40 will pass. Okay, then if 55% failure in phase 2, for example, then what we do, 40 into 55, okay, 22 will fail, so remaining will pass. Okay, out of 40, 22 fails, means remaining 18. Suppose if there is a 60% failure in phase 3, this 18, 60 into 18 will give you 10.8 failure, 7.2 pass. Okay, so if it is 60% failure in phase 1, 55% failure in phase 2, 60% failure in phase 3, only 7.2 molecules will pass through 100. So how do we, as a total, we can calculate 100 into 1 minus 0.6, 1 minus 0.55, 1 minus 0.6, that comes to 7.2. So a general formula, pass material is 100 into 1 minus P1 into 1 minus P2 into 1 minus P3, where P1, P2, P3 are failure probability in phase 1, phase 2, and phase 3 clinical trials respectively. Understand? Somebody had asked this question. I hope they understood this. So this is a basic knowledge which one can derive from their course in probability and statistics, which they might have studied in their first year training. Okay, so this is sort of a summary of what we have looked at in the past 3 weeks, in the past 15 video lectures. And I hope I will again meet you on another live session at just before the start of your online final certification exam. Okay, I hope you enjoy the course. Thank you very much for your time. Thank you.